I was able to get the first recording onto our Panopto site, so if you want to review that, it should be available for you. One other thing is, remember that when you look in those folders in the Panopto module, you look at the folders at the top, it says exam 1, 2, 3, 4. So you've got lectures that are recorded already. They're just the ones from the previous textbook. So it's the same content, but if you want to jump ahead or something like that and you want to view a lecture ahead of time, you can view those recordings. They're already available for you. Also included there are some what I call highlight reels in there. And so those ones are shorter introductory videos to each chapter that just kind of hits the highlights, the big topics, the more difficult things you want to focus on, etc. So like we were talking about before, when you are starting with a topic and you want to start simply and kind of just get some framework going, things like that, and the chapter summary, etc., those are good places to start so that you can see the major topics without all of the detail included, and then you can work your way up to things like the full lecture and reading the full chapter, etc. So those are also there. So when you go into each one of those folders in the Panopto, exam 1 or whatever, it'll say, blood chapter highlights or heart chapter highlights or whatever. So you can view those at any time too as an introductory little spiel into the topic at hand. Okay. So, whence last we stopped talking, we were here talking about erythrocytes. So we were into the cellular elements and we were looking at starting with the red blood cells. We're going to tour through white blood cells. We'll talk about platelets, the thrombocytes as well. And then we'll talk about the plasma. We'll wrap up talking about hemostasis and this whole clotting process and things like that. Okay? So one of the last things that we were talking about with the blood cells is we were saying that they're very specialized cells. They don't have a nucleus in the mature form. They give up a lot of their organelles. And so there are precursor cells that are being created during what's called erythropoiesis. So hematopoiesis is, the creation of blood cells overall, but erythropoiesis is specific to the erythrocytes or the red blood cells. So when we talk about the production of the red blood cells, you start with a stem cell where are these stem cells being generated? In the bone marrow, in the red bone marrow, right? So these precursor cells that we see here are inside of the bone marrow. And we start out with what's called a hemocytoblast. So this is a generic stem cell, right, that is going to give rise to later cells that are going to be red blood cells. So really what you need to know in terms of the different stages, you don't need to know all these different stages. You should know erythroblast, because remember we said the suffix, the blast, is usually a precursor form, like we talked about with chondroblast and osteoblast, etc. So you can learn that as kind of your earliest cell, the erythroblast in this erythrocyte development line. The reticulocyte is the last stage of the immature red blood cell. So the reticulocyte is where it's about to enter the bloodstream, and we can see that it's at the stage where it's giving up those organelles and maturing into the full-blown erythrocyte. So erythroblast, reticulocyte, and then finally erythrocytes are what are going to enter the bloodstream. Okay, so when we are looking at the numbers and the percentages, etc., we're looking for the numbers of the adult erythrocytes, if you will, or the full-grown erythrocytes. So when we look at it, we want that to be a certain percent. Like in lab today, we're doing hematocrit, so we want to make sure that there's about 45% or so. We do blood cell counts, and we look at the actual numbers as well. But because the function of these red blood cells is to carry oxygen, if you don't have enough of these, it could lead to both hypoxia as well as anemia. So hypoxia, when we look at that term, right? So this part, the prefix, hypo, means what? Right, low. So when you talked about the integumentary system, you had hypodermis, which is located underneath the dermis, etc. So hypoxia is low oxia, low oxygen levels. So if you don't have enough red blood cells to transport the oxygen, then you're not going to have enough oxygen being delivered to the tissues. So it certainly could lead to hypoxia, but also if you don't have if your carrying capacity for oxygen or you don't have enough red blood cells, we call that anemia. Now, there are different types of anemia as well, different types of hypoxia. We'll talk about that a little bit later on. But generally, too few red blood cells gives us anemia and hypoxia. Too many can go the other side. Poly, many, cell, right? Polycythemia is where you have too many. Now, in this case, you could have overactive red bone marrow or something like that. You could also, instead of having too many red blood cells, you could actually just have that ratio being off. Like if we don't have enough fluids and the plasma component is low, then that hematocrit is going to be off. It's going to have a higher hematocrit level and it'll look like polycythemia. So the amount of red blood cells in terms of the blood volume is too many. And the blood can become too viscous, right? So viscosity has to do with how liquid, how cohesive it is. So if you have something that's higher viscosity, sometimes you hear about it like in oil commercials or something like that. They talk about a high viscosity. That means it tends to stick together, right? It flows more slowly. It's like honey or oil or something like that. You can imagine you don't want something high viscosity trying to squeeze its way through these blood vessels, be ejected from the heart, etc. So it's a lot more stress on the cardiovascular system if you are polycythemic, if you have a high viscosity. So that actually can become dangerous. So in terms of controlling the amount of red blood cells that we have around and the production of them, remember it's called erythropoiesis, is the process of creating those erythrocytes. And there's a number of different kind of feedback mechanisms that the body monitors to make sure that the number of erythrocytes is going to be high enough. So oxygen levels is going to be a big one. We're monitoring oxygen levels. If those are getting too low, we need to kick up the production of red blood cells. But also, there are other factors that can affect how many red blood cells you have going in your body. So the amount of iron, the amount of amino acids, and even B vitamins can come into play here. We know iron's a factor for hemoglobin. Amino acids are what we use to create proteins. And so all those factors can come into play. So like we said, polycythemia, often too many red blood cells, increases the viscosity, and there are different reasons that can occur. So problems in the bone marrow. So if you have bone marrow cancer, right, cancerous cells are our own cells that are replicating out of control. There's too many of them, they're too active, etc. And so if that's occurring in the bone marrow, it's possible that they are doing their job there's too many of those cells and they're too active. So they're cranking out too many red blood cells. It's also possible, when you're talking about bone marrow cancer, white blood cells come from there as well. So you might see elevated white blood cell counts if you're looking at a blood count, like a differential, and that might have to do with too many white blood cells. And in that case, you are producing so many white blood cells that the red blood cell production gets pushed out of the way. So you might see anemia associated with that increased white blood cell production. Okay, so there's also what's called secondary polycythemia. That's caused by low oxygen levels. Now, in that case, that is something that naturally occurs. It's usually not a problem. Like, for instance, if you go and you visit family in Denver, Mile High City or something like that, and you're there for a couple weeks, the first week or so, sometimes you get very winded or you feel faint or something like that, and you start to exert yourself because there is less oxygen up at altitude, right? There's less atmosphere above you overall, so necessarily there's less oxygen as well. So you're struggling for a while to supply oxygen to your tissues. Your body responds to that kind of temporary hypoxia by jacking up the number of red blood cells that are being produced by the red bone marrow. So that will increase your red blood cell production and help in that process. Now, there's a lot of athletes that will do that as training as well. There's a lot of training facilities, Olympic training facilities, as well as cycling and things like that they utilize that natural feedback mechanism. Now, that's a natural way of increasing your red blood cell count. You have more red blood cells, you can deliver more oxygen to your active tissues like your muscles, so you can generally perform longer, you have a higher endurance, etc. Now, there are other ways that you can increase it, which is unnatural. So, for instance, we know this right here, EPO, is erythropoietin. That's the hormone that naturally comes from the kidneys The kidneys are monitoring oxygen levels, etc. When it gets too low, they release that hormone, goes back to the red bone marrow, increases red blood cell production. Okay? If you were to inject erythropoietin, EPO, then the red bone marrow is still going to get that signal. It doesn't know where it came from. It's just getting the message that we need to increase red blood cell production. Okay? So this has become a problem with athletes actually doing things, artificial things, to increase red blood cell production, to increase their performance. So it's banned by a bunch of different agencies, and it's also dangerous, right? It increases blood viscosity, like we said. It puts a lot of stress on the heart as well as the blood vessels. It's possible for people to have strokes and things like that. But there's still been various people that have been involved in blood doping. So Ricardo Rico was actually banned from the Tour de France because he tested positive for a erythropoietin-associated activator. And then this year, when we're looking at the Olympics, right? So there was a lot of the Russian teams that were actually either temporarily or for a longer duration banned from competing. So because they had been involved with different types of blood doping. And so there was actually a little kind of funny interaction you would see between some of our American swimmers, and then Yulia, you see over here, she was previously banned for blood doping, and then the Russians ended up letting her compete this year in swimming. And so each time after some people on our team would compete, and then Yulia would compete, they would get up and they'd be like, oh yeah, I'm #1. And then our team would be like, no, you're not. You did that artificially. It doesn't really count. So they would kind of go back and forth like that. But, so that's something that occurs naturally, but also can be influenced externally. So you do need to know, to be sure, erythropoietin. Now that polycythemia, you should know the definition of polycythemia and anemia. I wouldn't worry too much about that whole, bless you, polycythemia vera and secondary polycythemia. Don't worry about those levels of details, but you should know what anemia is and polycythemia. You should know what erythropoietin is as well. So remember that erythropoietin is a hormone, and it comes from the kidneys. So when we look at it, right, this is kind of the sequence that could come into play. So for instance, we enter into a state of hypoxia for whatever reason, and so we need to get back into a homeostatic set point. So what we're going to do is, those oxygen levels are reduced. It's sensed by the kidneys. They release that erythropoietin that we mentioned. That goes back to the red bone marrow, increases red blood cell production, and now we've gone back to our homeostatic set point. We have normal red blood cell count, we have normal blood oxygen levels, and everything is back to normal. Okay? Okay, so we're constantly making more red blood cells. We're monitoring the red blood cell levels that we have in the blood, and typically a red blood cell or erythrocyte is only going to last about 3 to 4 months. Remember, it doesn't have the cellular machinery to be able to maintain itself, to be able to maintain its plasma membrane, or produce proteins, or produce ATP in a aerobic fashion, etc. So there's a lot of wear and tear that they go through, and they end up dying off. When they die off, after that 3 to 4 months, they have to be removed. And so they're generally removed by these big macro eating cells, macrophages. And the macrophages themselves break down the red blood cells, but then oddly enough, even though these are red, we go through kind of a green process of recycling some of the components. So we try to hold on to some of the proteins, some of the iron, and then get it back to the red bone marrow so that we can make more cells again. So we talked about this part in terms of monitoring and maintaining red blood cell levels. We said that though when these red blood cells break down, we actually are going to try to harvest some of their components. So this is the hemoglobin. Hemoglobin is that protein that does what? It binds oxygen, right? So it's the whole way that the red blood cells are able to bind and carry oxygen. So we break down the hemoglobin. The globin is the protein part, and we break that down into amino acids that we can then use to synthesize other proteins. The heme group is broken down into 2 parts, bilirubin as well as the iron that we said is so very critical at the core of the heme group. And what we find is that the iron binds to a transport protein that's called transferrin, right? If you look at the periodic table, Fe is the chemical symbol for iron. Transferrin actually carries that iron around back to the red bone marrow so that we can use it. The heme group gets broken down into bilirubin. And bilirubin is actually what goes into bile, which is secreted, we'll talk about later, secreted and made by the liver, goes into the digestive tract. Bile has a number of different functions, but the bilirubin is something that we get rid of. And so the bilirubin is something that it's broken down further and contributes to the brownish coloration of poop. So that's where that coloration comes from. It comes from, right? Remember that these proteins and blood itself started off as red, and so red, brown, etc. It should be a brown itself. If you start seeing bright red in your feces, then obviously there's a number of problems that could be arising, including along this pathway here. But this is a way that we kind of recycle these particular red blood cells. So we try to get some of these raw materials back to facilitate erythrocyte production later on. Okay, so macrophages occur, like for instance in the liver, they occur in the spleen as well. We'll talk about the spleen later on as part of the immune system. But the macrophages are what take the red blood cells out to help to break them down so we can recycle them. All right, so we talked a little bit already about anemia, where we have low blood cell numbers or the oxygen carrying capacity is low for some reason. Now, there's lots of different ways that anemia can come about. Again, these you'll learn about more later as you get into your medical careers. I wouldn't worry about, in terms of the exam, I wouldn't necessarily worry about the different types of anemia, but if you look at them, you can see how these could arise. So for instance, hemorrhagic anemia, well, guess what? If a shark has bitten your leg off, you have automatically become anemic, right? Because you don't have the same oxygen carrying capacity in your cardiovascular system because it's spilling out into the ocean. So that's called hemorrhagic anemia. There's also hemolytic anemia, where the cells are lysing or basically rupturing too early, and so then your red blood cell count is low. Aplastic is where there's a destruction of the hematopoietic material or the bone marrow itself. This is one that you may run into yourself. It's called iron deficiency anemia. So this one is also called dietary anemia. So we know that hemoglobin is a protein. In order to make proteins, we have to have amino acids, we have to have iron, etc. Those are taken from your diet. So if you're not eating enough protein, usually animal protein, those are complete proteins, then your body doesn't have what it needs to be able to synthesize that hemoglobin that it has to all the time. So you might be able to make enough red blood cells, but the red blood cells don't have a high enough concentration of the hemoglobin protein to be able to carry oxygen. So that one is actually easily remedied. So you go to donate blood, they say that you're slightly anemic or something like that, and they recommend on a medical level that you go get a double it in and out. Generally, red meat, right, actually has higher blood counts in it as compared to white meat and plant proteins, etc. So it's an easy way to increase the amount of protein, the amount of iron in your diet, then that supplies your red bone marrow with what it needs. Pernicious anemia is another one. We said that vitamin D comes into play here as well. Vitamin B is important in terms of actually being able to create red blood cells, etc. So there are different ways that you can get into this low blood oxygen condition. You may have heard of this one as well, sickle cell anemia. So in this one, this is a condition where there's actually a change. Let me get over to here. We're not going to have a whole lecture on sickle cell anemia, but it's interesting because you hear about sickle cell anemia because the red blood cells, instead of having this normal biconcave disc shape, the cells take on this kind of sickle moon shape to them. And that has to do with a single amino acid change in the hemoglobin itself. So a hemoglobin is a protein made up of amino acids. It takes a specific sequence of amino acids to make that hemoglobin. And if in the number 6 spot you have a valine instead, then that throws off the entire sequence and the hemoglobin basically breaks down. The entire red blood cell doesn't maintain its shape, and so it can't carry oxygen as well. And they tend to degrade quicker, right? They tend to lyse quicker as well. So sickle cell anemia is a condition that results in low oxygen carrying capacity. Now, interestingly enough, oddly, it has a benefit as well. So people who have sickle cell anemia don't contract malaria. So in a way, it's not something you would necessarily want, but you have that perk where you don't get malaria there. So transfusions are usually how you would help to treat sickle cell anemia, but they're also looking into gene therapy because this has to do with an inherited trait where, again, when we're reading off the DNA, which is this big cookbook on how to make proteins, that recipe is corrupted. So using gene therapy, they're looking at the possibility of trying to get rid of that errant gene. Okay. In general, red blood cells out of our different cell types are some of the simpler ones. They do one job, basically carry oxygen. They have bunches of hemoglobin to help them do so. And for the most part, they all look the same. As we were talking about in lab this week, they're not exactly the same, but they look the same from the surface. If we go to our white blood cells instead, though, we see some differences. So white blood cells are called leukocytes, and the leukocytes are here to protect us, part of our bodily defenses. So the leukocytes, we know that we don't have as many of them. We've seen that in the microscope. In that same cubic millimeter where we have like 8 million red blood cells, we have maybe 5 to 10,000 white blood cells overall. So it's about 1% of the total blood volume. And when you're looking at a blood sample, when you're looking at hematocrit or something like that, those white blood cells are still there. On a microscopic level, if you were to use a dissecting scope and look at that capillary tube with your packed cell volume, there'd be a little white layer at the top of the red packed cell volume at the bottom. It's called a buffy coat. And that is where that little 1% ends up, right at the surface of it. But by your naked eye, you can't really see it because there's so many red blood cells. In any case, white blood cells have a totally different job in terms of protecting us. And very interesting thing, at least to me, about white blood cells is that they kind of have a mind of their own. So they can, rather than just being pushed around by hydrostatic pressure, blood pressure, etc. in the plasma, they can move on their own, and if they're doing their job, they are found more commonly outside of the bloodstream than inside of the bloodstream. As a security detail for the body, it doesn't make sense to have them only in the bloodstream, right? They need to be out amongst all the different cells and tissues, checking to see if we have a bacterial infection, if there are virus-infected cells, cancerous cells, or whatever. So they have to be out amongst the people, if you will. And so they can actually leave through capillaries and venules in a process that's called diapedesis. They can move. You guys heard of amoebas and stuff, right? Those are unicellular organisms that kind of move like a snail. They have like a pseudopod that they can extend out and then kind of pull themselves along. White blood cells move in the same way. So they'll be going along in the capillaries and the venules. They'll actually get a little cry for help, a chemical cry for help that says that there's an infection in that area. They will put the brakes on, and we'll talk about how they do that, adhere to the wall of the capillary venule, and then squeeze their way out between the cells of the vessel and start moving towards the site of infection. So, like for instance, if you, like we talked about, get that white head or something like that, right? You have some sort of pimple. Well, how'd those white blood cells get there? They were normally in the bloodstream. They've actually migrated from dermal capillaries and venules and then gone into where that bacterial infection is and have gone into that battle. Okay? So they can move on their own. So they move through the tissue spaces and they work to defend your body. Now, we said that usually there's 5 to 10,000 in a cubic millimeter of blood. You may see higher than normal counts go into what's called leukocytosis. And so with leukocytosis, you will see that if they are going into battle, right? There's a bacterial infection or some sort of pathogen. You're going to get more of them because, like with any war or battle, you need to bring more troops into it to win that battle. So we'll see artificially, if you will, actually it's natural, but you'll see an increase in the number of white blood cells. Okay? Okay, so we were focused here, where we have mainly erythrocytes. We said that leukocytes are also a cellular element, but only about 1%. And when we look at their count, if we're doing a differential, like we said, that you can do, we see that they separate into 2 different groups, granulocytes, agranulocytes. For the granulocytes, remember, they have the suffix -phil. So we have the neutrophils, eosinophils, and basophils. For the agranulocytes, they have the suffix "cyte," lymphocytes, and monocytes. So these ones, most common neutrophils, then lymphocytes, then monocytes, these are very rare, right? We did see some eosinophils, the ones that have the eosin stain, that stain pink. There were 1 or 2 people throughout all of my labs, actually this semester, we did see 1 or 2 basophils, but they are very rare indeed, like you can see from the percentages there. So we've already gone through this and seen them physically in the microscope and how they appear different, right? So things like our basophils and our eosinophils that we see there. Normally they're actually a little bit pinker than that in terms of our eosinophils. There's a little mnemonic to remember how the populations of the different white blood cells are distributed. Never let monkeys eat bananas. Seems counterintuitive, but it tells you that neutrophils are most numerous, then lymphocytes, then monocytes, then eosinophils, and then basophils. Okay? You don't have to memorize all of the distributions in terms of numbers, but you should know which are the most frequent. Like we said, neutrophil is definitely the most frequent. If you forget on a lab practical and it's pointing to one of these white blood cells, guess neutrophil, and you got a 50-50 chance. Okay, so we saw with the white blood cells that they all have nuclei. We said that we look at the shape of the nucleus, the size of the cell, whether it has granules to help us discern between them. And the neutrophils, these are basically a bunch of phagocytes. So these ones help to kill microbes in what's called a respiratory burst. What they do is they synthesize oxidizing substances. Think of things like bleach and hydrogen peroxide, how they help to kill off things, right? We're using bleach solutions to clean the tabletops in the lab. And so they also produce oxidizing substances in those little granules that we said that they have. So they will release those onto things like bacteria and help to kind of digest them and kill those microbes. They also have these things that are called defensive proteins that will pierce the membranes of these microbes and cause them to lyse. But these are phagocytic cells as well. So they basically will end up kind of attaching to and partially ingesting these cells. They secrete these digestive enzymes and proteins onto them and kill them off. And so for the most part, neutrophils are oftentimes known as the bacteria's bacteria slayers. So these are the body's bacteria slayers, and what they end up doing is handling, bacteria are pretty small, neutrophils of our different cells are kind of a medium-sized cell. You can imagine we have one other phagocyte that's a bit bigger than that. What's the biggest white blood cell? Monocyte. So that's another phagocyte, but it's going to handle larger cells. These handle mainly bacteria. So if we see an increase in the number of neutrophils, it may indicate some sort of bacterial infection somewhere. Okay? Okay, so neutrophils, eosinophils. Eosinophils, we said that they are red staining, they have granules, etc. And what they are specialized for are against parasitic worms, as well, they're associated with allergies. Now, if you have a tapeworm or something like that, these will release digestive enzymes again from their granules, which will be released onto the surface of that parasitic worm and start to digest the cells, the outer cells of the parasitic worm itself. So if you look at a differential blood cell count and you see this increase in eosinophils, then you start looking for some sort of parasitic worm in that particular person. You can also see increases, though, in eosinophils associated with allergies. They have a homeostatic mechanism working with the basophils to try to have an appropriate allergic response, where effectively you may increase blood flow to the particular area where you have this thing that's invading you, but we don't want to go full-blown. We don't want to be anaphylactic shock or something like that. And eosinophils will actually get rid of some of the molecules associated with immune responses, with swelling and that type of thing. And so if it starts to get too swollen and too inflamed, there'll be a release of or a taking up of some of these immune chemicals and proteins to keep that from going out of control. So sometimes you'll see eosinophil levels be higher in a blood cell count associated with allergies as well. Okay, basophils, like we said, also work in terms of, sometimes in terms of allergies, but also in terms of inflammation. Their main role is to contain and secrete histamine. So histamine basically increases the inflammatory response. So it makes you swell in that particular area. So when you look at that, histamine is going to be released. It's going to dilate blood vessels. It's going to make capillaries a little bit leakier. And it's going to allow our defensive cells to reach that site of infection easier. So if you dilate blood vessels, you're opening up the highway for our white blood cells to get there to fight that infection. Okay? So it increases inflammation. It's called histamine. Sometimes you hear about histamine. It seems familiar because there are medicines, right, that are called what? Antihistamines, right? So if you're getting too much swelling, you can take an antihistamine, which will decrease it. It decreases the activity of basophils and these other cells that are called mast cells, and it negates the effect of the histamine. So if you are swollen because you actually have an infection, right, you have a cut and it is red and swollen and hurts, well, that's a good type of swelling. You need that type of swelling because there's a battle going on there. Bacterial populations are getting bigger and bigger, so we need more of our defenses to go into that area, okay? You may have to help it out with, some sort of topical, kind of antimicrobial as well, but you need that type of swelling. Meanwhile, if you're talking about an allergy, we'll talk about this with the immune system when we get there, but an allergic reaction is where your body is responding to something as if it's a pathogen, like a bacterium or a virus or something like that. But instead, all it is pet dander or pollen or something like that, something that's really not a pathogen. It's not going to infect you and turn you into, a sunflower or a grass or something like that, but your body is overreacting as if it is. And so in those cases, if you have swelling, right, where are you being attacked? You're being attacked like at your eyes and your nose, and that's where you get your swelling. So histamine's being released there because, we're being attacked by pollen, right? So in those cases, you take an antihistamine because your body's overreacting and we want to decrease that swelling. Okay? So again, we'll talk about that in terms of bodily defenses later on. But basophils help with swelling in terms of increasing releases of histamine. All right. Agranulocytes, two other ones. We know that we have lymphocytes and we know that we have monocytes. Now, lymphocytes at this point, we're going to keep it rather simple. Lymphocytes help with what we call immunity. There's two types of lymphocytes, T lymphocytes and B lymphocytes, and they both help with the long-term protection against different types of pathogens. Okay? So they do it in different ways. B cells actually produce antibodies. T cells do a direct kind of cell-to-cell interaction and target virus-infected cells often and like cancerous tumors. But we have a whole chapter on the immune system where we will learn the details about T cells and B cells. So right now, as long as we know that lymphocytes are associated with immunity or this long-term protection from different types of disease-causing agents, that's okay for what we need to know about lymphocytes right now. Okay? Monocytes, on the other hand, we said it's our other type of big macrophage, and so or phagocyte, and so these ones are found in the blood, right, as monocytes, like we saw in the blood samples, but like other white blood cells, they can leave the circulation and go into the tissues. And when they do that, they get an additional name that is called macrophage. Again, macro just means big, phage, like to eat. So these ones are going to be eating bigger things. So for instance, things like virus-infected cells, as well as intracellular bacterial parasites, so different other types of parasites besides worms. So it's a bigger phagocyte that can handle bigger cells, like your own cells that have viruses inside of them. Okay? Okay, so monocytes effectively are the same as macrophages, but the macrophages are usually outside of the bloodstream as opposed to inside. Now, any of these white blood cells, we said, are going to be found in the blood, but the ones you find in the blood are just a small percentage of those that are in the body overall. Most of them are out on patrol. They're in the tissues doing their job, hunting for pathogens. So this is what I was talking about in terms of how they can emigrate or leave the bloodstream. Remember immigration, right? You hear about that, people wanting to get in or things wanting to get in. Emigration is leaving or exiting instead. So they emigrate from the blood to the site of infection. And what they do, The infected cells, or sometimes different types of proteins that are released from the bacteria or whatever, they put out a chemical signal. It's kind of like if you get home or something like that and somebody's cooking and it smells really good, you're like, oh my gosh, where are those cookies? And you just follow that scent trail, usually to the kitchen, and you can find out where it is, and now, right, you've gotten to your target. Same thing kind of happens for infected cells. They will release chemicals that are then picked up by the cells, like white blood cells, in the bloodstream, and it works as an attractant. So what they will do is they will start moving towards that site of infection in a process that's called chemotaxis. They are, if you will, taxiing towards those attractive chemicals. So chemotaxis, taxis is a very simple movement towards or away from some sort of stimulus. So for instance, plants are phototactic, and they're positively phototactic. So if you have a plant in a window and then you keep it alive long enough, which sometimes is a struggle when you're in college, but if you keep it alive long enough, oftentimes it'll end up bending over towards the window, right? You end up having to turn the pot because they're positively phototactic. Wherever the light source is, they grow towards it because they're getting the energy they need from it, right? There are other things which are negatively tactic. So for instance, if you go downstairs across from the lab, in the marine biology lab, we've got a number of different invertebrates in that lab. And a lot of them you don't see because they are negatively phototactic. If you look underneath some of the coral heads in there, you might see like an arm sticking out from some of our brittle stars, or some of our sea urchins. They're hiding when the light is there, because light to them means that, predators can see me. It also means, for a marine organism, that means it's potentially hot, like maybe it's low tide, and so now the sun's beating on me, I'm going to dry out or something like that. I need to be where it's dark, because it's going to be cooler there and it's going to be wetter there, and I'm going to have a better chance at surviving. So they have, even a brittle star, which is like a type of sea star, they have photoreceptors. They don't have eyes, but they have photoreceptors on their arms, which can tell them whether it's light or dark. They don't actually see images like we do, but it can tell light or dark. And wherever it's light, it's going away from. Wherever it's dark, it likes, and it's going to hide underneath there, because that's safety, because that's cooler, etc. So in any case, these cells are positively chemotactic. When these chemicals from the infected tissues are reaching them, that draws them to it. And so when they're going along, they get that chemical signal, they start to adhere to the wall of the blood vessel. That process is called margination. Molecularly, they're putting on the brakes and starting to stick to the wall in order to slow down, because they don't want to pass up the area where the infection is. So now they've attached to the wall. Now they go through this process of movement that's called diapedesis. So diapedesis is where, like you see in the figure there, they change their shape and they squeeze out between the cells in the blood vessel and then start moving like an amoeba towards the site of infection. Okay? So that's the way that they can be signaled to where they need to be to engage in that battle, and how they can physically move outside of that particular blood vessel. Okay, so this figure that we see here, this is from your book, and like the one that I showed you before, very good in terms of giving you all the information about different types of cells, what they do, like you see here in the last column, and then it gives you a little bit in terms of the frequency or distribution and what they look like. So that's a good thing to know, both for lab, right? We have to identify them in lab, but also in lecture. We said you could get a multiple choice question where we give you the function and you have to pick out the white blood cell that does that, like which is the one that helps out against parasitic worms. Or it could be like an essay question, again, where it says, okay, name 3 different types of white blood cell and say whether they're granulocyte or not, describe their function, that type of thing. Okay? Okay. So, white blood cells. Now, white blood cells, even though we have red bone marrow, which is where the red blood cells come from, white blood cells also come from the red bone marrow. Now, we'll talk about in the immune system, there is a possibility that certain types of white blood cells can actually come from other tissues later on as well, but for the most part, our white blood cells will come still from that red bone marrow. And when we talk about, there's that hemocytoblast you see in the second line there, the blood stem cell, that gives rise to both red blood cells and white blood cells. But what we find is that as we differentiate or specialize into these 5 different types of white blood cells, there's 2 different lines of stem cells. ones that are called myeloid stem cells and ones that are called lymphoid stem cells. So lymphoid stem cells become lymphocytes, right? So they have this particular cell line that develops only into lymphocytes. They have a big job in terms of protecting us and giving us immunity, like we said we're going to talk about later. So they have a dedicated cell line that are called lymphoblasts, which come from lymphoid stem cells. Meanwhile, the other 4 types of white blood cells all come from a different stem cell line. They come from myeloid stem cells as well. So if we look here, here's our hemocytoblast, myeloid stem cell, lymphoid stem cell. Follow that down, only become lymphocytes. Okay? Myeloid stem cells become all of the other cell types, both white as well. Technically, as we look along these pathways, there are some variations in there as well, but myeloid gives rise to everything else. Lymphoid directly go to lymphocytes instead. Okay. Now, sometimes you may have heard of this, right? Leukemia. So we've seen that the prefix leuc and leuko is talking about the white blood cells. And in this case, what's happening here is you may have cancerous cells within the bone marrow of these stem cell lines. And so what's happening is they are cranking out more and more and more and more white blood cells, more than you need. And like we said, it's actually crowding out some of the erythrocyte cell lines. So it becomes totally occupied with these cancerous leukocytes. And so we see extremely high numbers of white blood cells. We become anemic because of the lack of red blood cells, etc. But the problem is that this is stem cells, so it's giving rise to all of these cells that protect us, etc. And so it's very, very problematical in terms of the body because all of there are different types of body systems can be affected. There's other different types of diseases and disorders. You hear about this, right? Mono. So mono, what happens is you get artificially high agranulocytes, and some of those lymphocytes as well as the white blood cell counts go up, particularly the monocytes, which is where you get the name in terms of mononucleosis. But this is a viral disease that can be passed on. So that's why you hear about it as the kissing disease. Okay, red blood cells, white blood cells, wrapping around to platelets. So platelets, we said, are not whole cells. These ones are fragments of cells. The precursor cell for those are really big cells called megakaryocytes. They also come from the red bone marrow. But the interesting thing about it is that these megakaryocytes, here's your hemocytoblast again, and then here you see how it develops into a megakaryocyte. The megakaryocyte is so big that it can't exit the bone marrow and go into the bloodstream intact. So what happens is, as it is exiting the red bone marrow, it basically goes out like shredded cheese, right? It goes through the grater of some of the pores of the red bone marrow, entering into the bloodstream, and it breaks it apart into all these cell fragments that we call platelets or thrombocytes. So the precursor cell is a megakaryocyte, a very large cell, but then it gets shredded along the way, exiting the bone marrow, and becomes all these cell fragments that we call platelets or thrombocytes. So why do we need all these broken pieces of a megakaryocyte? Well, the platelets have a role in terms of our clotting cascade and basically helping us to maintain the amount of blood that we have inside of the cardiovascular system, or what we call hemostasis. This figure here I interjected just because it shows the development of all of our different cell types. There's our megakaryocyte again, giving rise to our platelets or thrombocytes. But remember, look over on the left-hand side here. Myeloid stem cells, all of the 4 different types of white blood cells except for lymphocytes, and it gives rise to the platelets and the red blood cells. So myeloid stem cells differentiate into all of our different blood cell types, except lymphoid stem cells handle our lymphocytes, whether T or B lymphocytes. Okay? So again, don't worry about a lot of these other intermediates like you see here, just kind of some of the ones, the box ones that I show you here. Hemocytoblast as your stem cell, myeloid or lymphoid stem cells, and then some of these intermediates that I have boxed up here, megakaryocyte, reticulocyte, etc. Okay? Okay, So we will certainly talk about the role of our platelets in terms of hemostasis coming up. But each one of these different types of blood cells are going to be carefully controlled so that we make sure we have enough in the bloodstream at any particular time so that they can do their job. And so there are always going to be what are called these hemopoietic growth factors that stimulate the bone marrow to make more of these different types of cells. We said it was EPO, or erythropoietin, for red blood cells. For white blood cells, there's different types. So collectively, we have a group of stimulating factors called cytokines. Cytokines will selectively increase the production of our different types of white blood cells. So cytokines are the chemicals responsible for growing more white blood cells. And then for the platelets, remember those are called thrombocytes. So the stimulating factor for those are called thrombopoietin. So thrombopoietin is a hormone from the liver that stimulates the red bone marrow to make more platelets. So we can control all of those separately. And that's important because they all have separate jobs. Okay. So one of the last topics that we get into in terms of the blood has to do with how do we maintain the blood overall, and this thing that's called hemostasis. Before we do that, though, do you guys have questions about anything that we've been talking about so far? Particular cell types, where they come from, etc.? Yeah,
One of the slides said B cells diversify themselves with texture.
Oh, so yes, so when we were talking about lymphocytes give us this long-term immunity, like we were saying. T lymphocytes and B lymphocytes protect us in different ways. We said that T lymphocytes basically do this cell-to-cell interactions. They attach to and attack like virus-infected cells and secrete these nasty chemicals onto them and cause them to die. B cells, on the other hand, what they do is they morph into a cell that's called a plasma cell, like you saw in some of the diagrams there. And what they do is they release antibodies. So when we're talking about antibodies, much like the antibodies that we saw in terms of blood types and things like that, those proteins can be secreted to defend us against all different types of things, like we said, like bacteria, etc. So they will adhere to different types of bacteria. So those antibodies that are floating around in our blood can attack bacteria and other types of pathogenic cells, and it's the plasma cells or the B cells that put those out. So antibodies are produced by our B lymphocytes. Other things? Yeah?
There was one slide that was covered by a picture on neutrophils, like in the printout.
Oh,
And I was just wondering,
yes.
when you go and look on Canvas, you should be able to see the slide without the picture.
Right.
Oh, it's printed.
Yeah, that's true. So that's the one with the...
Or
Yeah,
about the
you
note.
have the neutrophils select enable editing, and then you can drag it out.
Yeah, sorry about that. Sometimes when I have other things popping up there, then when you print them out, like in terms of the notes, then it automatically shows all the different layers. But if you go to the actual PowerPoint presentation that you have, when you download it, you can kind of click through it. And then as you click through it, you'll actually see those things and they'll come up. So yeah, you download it and then you click enable editing. So you could, in theory, make up your own slides and stuff like that. But you can also click through it like I am and see those things out of the way and in the way, etc.
Besides macrophage, what are the, is there more than one besides macrophage that does the phagocytes?
So the neutrophils technically also are phagocytes. So they're just smaller ones. So they end up eating and digesting bacteria like we talked about. In most recent times, they have found that eosinophils can also have a type of phagocytic response as well. But that's something they've just discovered like in the last 5 to 10 years. Yeah. Okay. Other things. Questions. All right. Okay. So we need to get into hemostasis, how we can stop bleeding, how, if blood cells are leaking out of the cardiovascular system in a way that we don't want them to, how can we stop that? And how can we get back to homeostasis in terms of blood? So for hemostasis, right, hemo blood, not homeostasis, the type of homeostasis. For hemostasis, there's 3 different phases. So if you have a cut blood vessel, how do we, number 1, stop that leakage from all over the place? Number 2, how do we get back to having a nice normal pipe there. You know, if we look at the blood vessel as like a pipe conducting fluid. So when we talk about hemostasis, there's 3 phases. The first one is called vascular spasm. The second one is platelet plug formation. So now we're getting an idea about the role of platelets in terms of hemostasis. And the third stage is actually blood clotting or coagulation. Remember, coagulation different from what we were seeing in blood in the blood lab when we were doing blood typing. When we see that clumping is agglutination. This is coagulation, a whole separate process in terms of clotting, forming of blood clots or scabs or whatever. Okay, so 3 steps: vascular spasm, platelet plug formation, and then coagulation. Now, vascular spasm is pretty straightforward. If you think about a spasm, right, it's a movement. It's like an uncontrolled movement. And so it's going to require muscle. We haven't gotten into blood vessels yet, but you probably know there are arteries and veins. There are capillaries, the smallest ones as well. There are also some medium-sized blood vessels that are called arterioles and venules. So capillaries are super duper thin. They're 1 cell layer thick in terms of epithelial cells, which makes it easy for things to get across their walls, which is good. Oxygen and nutrients and water, etc. The larger blood vessels have 3 different layers to their wall. They have an epithelial layer lining them on the inside. They have a connective tissue layer on the outside, and then they have a smooth muscle layer in the middle. So if it's a blood vessel, a larger one that has smooth muscle in its wall, so not a capillary, then if it gets cut and it's doing this like a fire hose, we want to do what we can. Like if you have a hose that's going in your yard and it's spraying all over the place, if you can't make it to the spigot to turn it off, one of the things you can do temporarily, you grab the hose and you kink it, right? So you kink that hose, now you've restricted the diameter of it, and there's not as much that's leaking out the end. So the blood vessels, at the very onset, if they have that smooth muscle layer, they try to do the same thing. They try to start constricting and decreasing their diameter so that there's less blood leaking out, okay? So vascular spasm, they go through vasoconstriction in order to try to decrease the amount of blood being lost. Vaso, a lot of people look at that and they go, oh, vein. Vaso just means vessel, so it could be a small artery like an arterial or whatever. Now, if you're talking about, as we come into horror movie season, if you're talking about a cut jugular or a carotid artery up here that has a diameter like a quarter, that's not going to do the job, right? It may try to do something, but it's still just going to be like hitting the wall, right? So that's mainly for things like arterioles and venules, etc. So vascular spasm, first step. Second step is platelet plug formation. So again, much like if you have a pipe broken at home or something like that, and you see a bunch of fluid coming out, your first instinct is to just put something over it, right? Or stuff something in it so it's not shooting out quite so bad. Then you're trying to figure out, okay, now what do I do in terms of actually repairing it? I can't leave it like that. That little rag that I stuffed in there, that isn't going to hold. What do I do? So same thing in terms of hemostasis. Second step is jam something in there. We don't have a rag, but we do have a whole bunch of these fragments floating around that we call platelets, and those will automatically start adhering to and jamming themselves into that open space. Okay? So what happens is, like we said, with a blood vessel, normally with a blood vessel, you've got an epithelial lining on the inside, then you have the muscle layer, then you have connective tissue on the outside. So as the blood's going through it, it normally just interacts with that endothelial or epithelial lining. That's what it sees on its day-to-day basis. Now, if there's a break in the wall, now all of a sudden, that blood is coming into contact with smooth muscle or connective tissue on the outside. Connective tissue with collagen fibers in it to reinforce it. Those collagen fibers end up being the stimulus for this clotting cascade or this platelet plug to begin in terms of sealing off that hole. So if the blood gets exposed to those collagen fibers, the platelets automatically start sticking to them and sticking to anywhere around we have that break. So it ends up being the stimulus to them to start forming this thing that we call a platelet plug. Now, once they start sticking to it themselves, what they do is they start releasing a variety of different chemicals. And you look at those chemicals, you're like, wow, I didn't realize those were used for this too, right? Serotonin, you hear about in terms of a chemical messenger in other areas. ADP, that's adenosine diphosphate, not triphosphate. That's the broken down form of it. But those chemicals are released from the platelets. They actually draw in other platelets. So now this is the beginning of that positive feedback loop. We see more and more and more responses occurring to try to plug up this hole that's occurred in the blood vessel itself. So the platelets stick to the wall, they end up sticking to each other, and they form what's called a platelet plug. I thought I had a picture of it. There's probably a picture coming up. But now we've jammed these platelets into the hole. That's a good first step. But we know there's a lot of blood pressure in these blood vessels. It's not going to hold. You jam a rag into that pipe, that's a good short-term fix, but we need to repair the pipe. So after the platelet plug, we get to the third step, which is actually clotting or coagulation. Now, like we said, this is a little bit more complex as opposed to just platelets jamming themselves in there. In this case, what we have is we're actually turning components of the blood from kind of a liquid soluble form into a solid form to form a scab or a clot or something that's going to cover up that hole. So what we find is that there are three main steps to this reaction. And so these are chemicals that we find in the blood that will, some of them, there's a whole sequence. A lot of them are proteins, some of them are enzymes. But what happens is, if you look at it, we can see that there are activators here. So we see this prothrombin here gets activated into a molecule that's called thrombin. The thrombin itself catalyzes, right? Enzyme catalyzes the conversion of this molecule, this protein here called fibrinogen, into fibrin. Now, when we were introduced to what components of the blood we had a long time ago, we said that floating around in the plasma, we've got a bunch of different things. There's ions, and there's hormones, and there's molecules. We said there are three different plasma proteins that are always floating around in there. Albumin, we said, is important in terms of osmotic pressure, so that we can transfer water back and forth from the tissues. But we also said that there's one in there called fibrinogen. So fibrinogen is always in the blood, and it's soluble. It's always there, but it's in a soluble form until this chemical cascade activates it and turns it from the soluble form called fibrinogen into an insoluble form called fibrin. And as the name indicates, it starts to form these fibrin threads. It drops out of solution and starts to form these protein threads. Think of it, right? We already have that platelet plug there. Think of it as kind of rebar in concrete, giving it extra strength, or even like a spider web that's going to attach to the platelet plug and work to reinforce it, okay? So these are our different steps like we were talking about. Vascular spasm, if we can, platelet plug, like we see, adhering to the broken walls of the blood vessel, as well as to each other. And then finally, we're going to reinforce that with coagulation. When the fibrin gets activated, it's going to reinforce it and form a clot on here as well. So that's going to be more substantial to be able to allow the blood vessel to still transport blood, but keep it from leaking out here. Now that clot is going to be solid enough to maintain the wall, but it's also going to give us a chance to rebuild the wall of the blood vessel so that ultimately we can get rid of that clot, open up the blood vessel in its entirety again, and have it back to its normal state where it has a wall that's all healed up. Whether it's collagen fibers and kind of scar tissue, or whether it's functional like epithelial tissue, etc., we want to get back to having an intact blood vessel again. So having the clot there not only stops the blood from leaking, but it gives the blood vessel time to heal again. And then, once the blood vessel is healed again, we'll actually get rid of that clot through a separate process. Okay, so we said that this clot is formed through a chemical cascade. So there's a series of steps involved here that ultimately result in the activation of fibrinogen into fibrin, which then reinforces the clot, like that, right? So there's two different ways that this can be activated. Ultimately, like we said, we have prothrombin, which is associated with platelets, right? Converting to thrombin. Thrombin converts the fibrinogen into fibrin. Now, there's like 13 different factors in this chemical cascade. It's like a whole series of dominoes. This activates that, etc. Okay? And that whole sequence has to occur in order for fibrin to be formed. But what you'll see is in this figure, there's actually two different ways that we can get to this endpoint where fibrin is activated. One is called an extrinsic pathway that you see along the left, and the other one is called the intrinsic pathway that you see along the right. So the intrinsic pathway, when you see in, that's trying to form a clot inside of the blood vessel itself. So this is a clotting cascade that is helping to form a clot inside the blood vessel, but oftentimes there's going to be some damage to the surrounding area as well. And so we want to kind of seal off that area and form a clot or a wall, kind of like sandbagging around that area as well. So that is what's called the extrinsic pathway, exterior to the blood vessel. So with these pathways, both of them lead to the formation of the fibrin threads, like we mentioned. But the intrinsic pathway actually goes through the full sequence of events, all 13 different factors, etc., getting to it, whereas the extrinsic pathway takes a shortcut. It's only a certain number, a reduced number of activators in order to get to the plasma protein, fibrin, being activated. Okay? So with these ones, the intrinsic pathway, like we said, because it's occurring within the blood, extrinsic pathway is going to be started with factors outside the blood with the surrounding tissues. And the extrinsic pathway, like we said, bypasses a number of those steps. So that, like we said, like if you, like in Baton Rouge recently, right, when the levees are breaking and the water's going everywhere, we want to, it's already gone over the banks of the river. Now what we're trying to do is save the home. So we want to sandbag out here to make sure that it's not continuing to flow even further. So we got to do that fast. That's what the extrinsic pathway is doing. It's still clotting cascade, it's still forming fibrin threads, but it's doing so in a quicker fashion so that we can make sure that this isn't spreading even further. Meanwhile, the intrinsic pathway is doing the same thing, but with the whole series of steps and forming that clot in the inside of the blood vessel instead. Okay? So, like I said, you don't have to know all these different factors, like 3 activates 13 and the Hagman factor and blah, 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 blah. Don't care about that. Okay? You should know that there are 2 different pathways, extrinsic and intrinsic. You should know, where they're occurring, and you should know that both of them end up activating the fibrin. It's just the extrinsic is faster, has fewer steps, and is occurring outside of the blood vessel. Intrinsic utilizes all the steps and is occurring inside the blood vessel instead. Okay? Okay. So what we find, we have formed these clots either inside of the blood vessel or outside of the blood vessel. Now, you may have learned in 201 that clot formation is one of the few examples in the body of positive feedback. Most cases, when we're talking about homeostasis, we're talking about negative feedback, right? So you have a set point, and we want to nudge it back towards the set point all the time. So if it starts getting away from the set point, we nudge it back. And then when it starts getting away, we nudge it back, right? Any change that happens, we have a negative response, a response that's opposite to it, pushing it back. But positive feedback, if there's a change that happens, that change gets amplified, so it changes more and more and more. So that doesn't maintain homeostasis very well. But in the case of forming blood clots, it's a good way to form a blood clot very quickly so that we don't have blood leaking all over the place. So once this process starts in terms of forming fibrin, having platelets stuck to it and fibrin threads attached to it, which actually, like we said, end up like spider webs catching red blood cells and other things into it, that just increases and increases and increases. It makes it bigger, it makes it more solid, so that blood vessel isn't going to blow open again. But if you're thinking about it then, you're like, okay, we made this clot. There are steps where it actually tightens. It actually gets rid of some of the fluid, and the cells actually grab each other and can pull in tighter. That platelet plug, those platelets actually have, when you were talking about the muscular system, actin and myosin. What do actin and myosin do? They help to contract, right? It's that whole sliding filament mechanism. They move the cell. Platelets actually have actin and myosin in them as well. So once they're attached to the wall of the blood vessel and they're attached to each other, they can pull together and actually tighten that clot and actually pull in the walls of the broken blood vessel so it's a little bit smaller. And so that's called clot retraction, that they kind of tighten in that way, squeeze out some of the excess fluid and make a nice solid clot. So we have ways of making this, of tightening it up, but at some point, we're going to have to get rid of it as well. So there is a process where we remove these clotting factors soon after they arrive there. So they do their job and you kind of sweep them out so that they're not really making it form too quickly. And then also, we have a way of inhibiting activating clotting factors as well. We'll find that there's one more protein in the blood that is critical for getting rid of blood clots, because we don't want it to stay there forever. We just want it there until the blood vessel heals, and then we want to get rid of it. So what we find is that there is another control mechanism for blood clots that works to get rid of them, that works to lyse or digest the fibrin and the clot and get rid of it. So there's two reasons we would want to do that. Number one, if we have a clot that has correctly formed because we have a broken blood vessel, at some point we need to get rid of it. So how do we do that? But number two, occasionally you may get a clot that is formed inappropriately. So you hear about things like DVT, right? So DVT, deep vein thrombosis, thrombocytes, platelets. Okay, so now I'm seeing there's some sort of link there, right? So there are various ways that clots can sometimes form erroneously. So for instance, you hear about if you get blood vessels that start accumulating fatty plaques in them, like in older people, those fatty plaques that end up adhering to the walls of blood vessels and narrowing them, that's a problem for obvious reasons, blood pressure, blood flow. But when those fatty plaques end up attaching to the wall, sometimes it sets off the clotting cascade. Because normally, like we said, they're just going through and looking at epithelia. Now it's like, what is this stuff? That stuff shouldn't be there. Maybe I'm supposed to stick to it. And the whole clotting cascade starts forming on top of the fatty plaque and makes it worse. They also can form sometimes just if the blood flow starts slowing down, right? Normally, the proteins and the platelets are whipping through there and circulating around the entire body once per minute. But if for some reason it starts slowing down, it's almost like these different clotting factors and platelets kind of get bored. They're like, well, I've been here for a while. Why don't we just start sticking to stuff, right? So if, for instance, like if you're in the hospital and you're not moving a lot, or if you're on a long flight or something like that and you keep your limbs kind of bent, it restricts blood flow. And now, especially in veins where it's already somewhat slow, now it has to go around that corner, and so as it slows down too much, sometimes erroneous clots will start to form at those bends like that. So that's why if you're in the hospital or something like that, they have you do different exercises, or they have you get up and walk around. And if you're taking long flights, like to Australia and it's 22 hours or something, they tell you to do some leg exercises, to get up and walk around to increase the blood flow and make sure that those clots aren't forming. So regardless, in that case, if we form an inappropriate clot, we've got to be able to get rid of it as well. So there are some mechanisms where we can lyse it, where we can dissolve it, utilizing some different factors that are inside of the blood. So this one here is one of the most important factors. This one is called plasmin. Now notice it has an inactive form called plasminogen and an active form called plasmin. We've seen that kind of way that we name things, like we saw with fibrinogen, right? Fibrinogen inactive form, when it becomes activated, we call it fibrin. So we take off the O-gen, and now it's the active form. Same thing here for plasmin. So this mechanism works to dissolve clots. And the cool thing about this, I think, is that the plasminogen and the fibrinogen are both floating around in the plasma. They're ready to do their jobs at any particular time. But whereas the fibrinogen activation process is very, very fast so that we can form a clot and we don't bleed out all over the place, the plasminogen activation process is very slow. So it's like the whole tortoise and the hare. They start out at the same point and they end up at the same point, but generally the tortoise is going to do it in a much slower pace, right? So we activate both of these proteins at the beginning of this clotting cascade. The fibrinogen forms fibrin very quickly. We form the clot. It's around for a while. By the time the clot has been around for a long time, plugging the hole and letting the blood vessel heal, by the time the healing has occurred, now finally that whole plasmin activation process has finished. Now the plasmin's activated. It actually starts eating away at the blood clot. And it's the perfect time because the clot's already been around for a while, the blood vessels had a chance to heal, and it's actually time for us to get rid of that clot. So they both get activated at the start, Fibrinogen does its job very quickly. Plasminogen takes a long time to activate, but when it does, then it's going to help to digest those fibrin threads, break it into pieces, and then we have macrophages and things like that that can work to get rid of those different parts of the clot. So it's pretty cool that it actually has this process of getting rid of it. Otherwise, right, positive feedback, clot would just get bigger, 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 and totally end up plugging up that blood vessel. Okay, so platelets involved in blood clots helping to initiate this whole chemical cascade, okay? One other thing related to platelets that's kind of interesting is they found somewhat recently, this is from a news article that came out just a few years back, they found out that platelets can actually attack malarial-infected red blood cells. So we said that, right, if you have sickle cell anemia, you can't get malaria, but if you do get malaria, if you get infected, they found that platelets have a secondary role in terms of helping to aid our body in terms of this parasite that has infected you. So they start adhering to and attacking the infected red blood cells and trying to get rid of them. It's something they didn't know that platelets could do. The main role, platelet plug, etc. But now they're finding out that they can do other things too. So there's always something new to learn in anatomy and physiology, to be sure. Okay, so next time what we're going to do is we will finish up blood and we'll be moving into the next chapter, which is heart. So make sure that you have downloaded or accessing the heart chapter information because we'll be jumping into that when we meet again.